Thanks for the introduction. Well, we've heard a lot yesterday and today about how increases in CO2 concentrations increase carbon uptake by plants. Well, the same is true for leaves. So as you increase the CO2 concentration, you tend to get an increase in CO2 uptake in leaves. And especially at lower CO2 concentrations, you get a stimulation of CO2. And as you move towards higher CO2 concentration, that becomes less and less. So we can take those measurements and fit them to a mathematical model that sort of describes the biochemistry behind that. And if we then know the variables that go into these models, we can then predict photosynthesis along the CO2 gradient, for example. If we then know how these variables change with the environmental variables, such as temperature, light intensity, CO2 concentration, and so on, we can then start to predict photosynthesis or estimate rates of photosynthesis across space and across time, and then eventually come up with maps like this that predict gross primary productivity even across the globe. So there's one slight issue with this, which is the concentration going into these models is the chloroplastic CO2 concentration, because that's where Rubisco sits that actually assimilates CO2. And why is that an issue? Well, schematic here, we have CO2 on the outside of a leaf, and that CO2 needs to diffuse through the stomata into the intercellular space, and then onwards across the cell wall and the membranes into the chloroplast where Rubisco sits and actually assimilates that CO2. And the stomata provides a diffusive resistance, which causes the CO2 on the inside of the leaf to be lower than out here on the outside. And similarly, these cell wall components and so on provide a resistance that impair that CO2 diffusion. Well, the consequence of that is that the chloroplastic CO2 concentration is significantly lower than the CO2 concentration in the air. Well, we have an easy way to account for this drop in CO2 concentration across the stomata, because we can use transpired water to approximate that. But it's much more complicated for this mesophyll diffusion resistance. That means in practice, in general, most people forget about this chloroplastic part and use the intercellular CO2 concentration as a proxy. Obviously, that causes some error, and to account for that, generally we use apparent values of those physiological variables. So not the actual true values, but apparent values. So that works on average. So let's say where the plant usually operates, that comes out with a good value. But we get a consistent bias and overestimate CO2 assimilation rates at low CO2 concentrations, and we underestimate CO2 uptake rates at higher CO2 concentrations. And that's obviously a problem if you then try to predict CO2 uptake as we go in the future, where we consistently have a higher CO2 concentration, and therefore tend to underestimate our CO2 uptake. So this recent paper here by Knauer et al., they tried to quantify that on the global scale. So they modeled GPP explicitly accounting for this mesophyll CO2 diffusion relative to this fudged approach where we use this apparent value based on the intercellular CO2 concentration. And as you can appreciate here, this is mostly red, which corresponds to 10 to 20% difference in GPP that we would predict by ignoring mesophyll diffusion. So how do we get about this? Well, luckily there's some way that we can use to estimate this mesophyll diffusion, which is make use of the abundance of natural carbon isotopes in the air. So we have heavy 13C CO2, which is the heavy CO2 in the air that occurs naturally, which diffuses slower into the chloroplast, is also being metabolized by biochemical reactions slower. And that means the plant preferentially takes up this light CO2, the C12C2. And as a consequence from that, the heavy CO2 is left behind. And we can measure that. So this is the discrimination, the apparent discrimination of the plant against the heavy CO2. And about 40 years ago, Farquhar et al. Came up with this model that sort of attributes this discrimination to different components. So we have a component in this model that accounts for the diffusion across the stomata, a component that accounts for the diffusion across the mesophyll, and then a component that relates to the biochemistry of rubisco, and then lastly, two components relating to the C2 being released from photorespiration and respiration. So this model has become probably the most used model in all of plant sciences. So it's very successful and can be used for many different purposes. But despite its popularity, when I started using that, I found that it often returns very unpredictable values and is not actually, in many cases, not very useful. So I set out to test this model and see if it actually is an appropriate thing to use. And the idea behind that was, if you measure this mesophyll conductance value with slightly different isotopic concentrations of the measurement gas, we would expect that mesophyll conductance should remain the same. So we should get the same value out of that. But it turns out, so these are measurements of mesophyll conductance with two different gases having two different isotopic compositions at a range of CO2 concentrations. And you can see that as you go to lower and lower CO2 concentrations, those two measures of mesophyll conductance diverge further and further, to the point where you even get negative values, which doesn't really make sense physiologically. So we can, for one thing, that this model has some issues that well, prevents us to get proper values of mesophyll conductance. We can also point to this respiratory component here as the culprit, because this is the only part of the equation that actually makes use of that isotopic composition of the measurement gas. So let's go slightly bit deeper into the assumptions going into the model. So really, it's the CO2 fixation by rubisco, which might fractionate, which adds to a pool of carbon that is being assimilated. And from this pool, we get respiration and photorespiration, which both might fractionate, drawing carbon. So that means if respiration, say, uses preferentially the light CO2, then heavy CO2 is left over in this pool of assimilated carbon. And that also means that the photorespiration in turn becomes heavier. So is that actually a reasonable assumption? If I just pull up this textbook example of respiration, where we take pyruvate, decarboxylate it completely in the TCA cycle to generate energy, then that means that there shouldn't be any fractionation, because the entire pyruvate carbon ends up in CO2, and this fractionation should disappear, and we wouldn't have a problem. But we do have a problem, so this can't be the situation. So instead, maybe we should assume that the TCA cycle is actually there to provide metabolites for other biochemical processes, such as amino acids, fatty acids, and so on. And these can't really be converted back to carbohydrates. So what I was proposing is, well, we should have a second pool, the carbon that's left over from respiration, these amino acids and so on. We should assume that they can't be converted back to carbohydrates and can't mix there. So if you then go back and derive the model from scratch using this assumption, we end up this equation here. And I excuse you if you think, oh, that's exactly the same, because it looks very much the same as what they had before. There's one important difference, which is that in the new model, respiration is scaled with the assimilation rate, whereas in the old model, it's scaled with the carboxylation rate. And that is important as you go for assimilation towards 0. Then this term becomes infinite, whereas that term down here stays small. And infinite is a very big number, so you really need to get this right. So if you take the same data that I showed you before, and just as a clarification, the assimilation rate against C2, when you measured, when you displayed it that, was chosen that assimilation rate goes towards 0. So the idea was to test what happens when this term becomes very large. Well, if you then calculate mesophyll conductance, suddenly the mesophyll conductance values you get with these two measurement gases are virtually identical. So now we have an experimental test that this model here is actually behaving much more robust and returning much more accurate values or sensible values. Which also means now we have really for the first time a way to accurately estimate mesophyll conductance and see how it varies with environmental conditions such as CO2, light intensity, temperature, and so on. And the idea then is to use that as a way to unfudge, if you will, things like the carbon uptake that goes into those global models to come up with a better prediction of the global carbon cycle in the end. And thanks to NERC, which has promised to fund this research, I'll be doing some work out at BIFoR Phase 2, sort of get this started. And that just leaves me to thank Misha, Graham, and Hillary, who were partners in crime on this modeling exercise. And thank you for your attention.
Sorry, wait for my microphone to go. Plenty of time for questions there. I'll take one from online if we're waiting in the room for people to come up with ideas.
So there is a question from Thorsten Grans. So in the map of Knauer and al., the effect caused by neglecting the difference between CI and CC was rather small for the Amazon region. Why is this? Is this related to temperature?
That's a good question, and I don't know. I would refer to Knauer et al., who probably have a better idea of what the driver is. Yeah, the short answer is I don't know. But yes, there's spatial variation in that.
And Jerome?
It's really, really interesting. Thank you. Out of curiosity, is there a direct method to measure mesophyll conductance not using isotopes?
There's other ways to measure mesophyll conductance, but none of them is direct. So you can use the combination between chlorophyll fluorescence and gas exchange, for example, which has its own assumptions going in there. You can use curve fitting of CO2 uptake data, which has another set of assumptions. And you always end up with slightly different values. So yes, there's other ways around, but none of them is direct. And in the end, it turns out that the new isotope discrimination model actually better aligns with the other methods to end up with similar.
Okay, and a backup question then: what is the validation of the data set? How do you verify all these assumptions?
Well, I mean, you can compare those different methods. In the end, getting a true absolute value is going to be very difficult in any case. But at least now we know the model behaves the way it should be. And if you don't know the parameters going in exactly, you end up with slight variations in mesophyll conductance. But that would be, I would say, mostly due to the uncertainties in the parameters and less to uncertainties in the model that we use.
Thanks.
Another question from online. Any other in the room? Yeah, Bruno?
So I'm just thinking about all of the water use efficiency estimates that people have been doing with tree rings and considering CO2 concentrations from the past. After you changed the model and included the assimilation parameter there, then you had much more reliable predictions. So how much does that influence the current water use efficiency change estimates that we have?
I'd say it depends. So this value here is ambient CO2, and the two values you get for mesophyll conductance at ambient CO2 when assimilation rate is reasonably high are very close. So the impact of respiration, just because respiration is small compared to A, is going to be small. The lower assimilation rate gets, the bigger and bigger the effect is going to become. So if stomata close, let's say, your effect is going to be huge. But the lower assimilation rate gets, the bigger and bigger the effect is going to become. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. So if stomata close, let's say, your effect is going to be huge. but under ideal conditions, it's not so huge. So I guess it depends on what conditions the plant is operating in. And especially if they close the stomata, then you would get a big impact, I think. Under ideal situations, maybe not so much.
Thank you, Florian. I think we'll move on from there. So, and next up,
No, definitely not.
okay, while Bridget's getting her
Is
microphone,
this working now? No, it doesn't seem to be.
he'll turn you on in a second. It's okay. So next up, we have Bridget Warren, who's one of our PhD students at BIFoR. And take it away, Bridget.
Oh, heck, I forgot my timer. Oh, well. So, hi, thank you very much for the introduction. And yes, I'm a PhD student here at Birmingham, and I'm looking at BIFoR FACE, but I'm actually a geologist in the Department of Earth Sciences. And Florian was just talking about carbon isotopes in general in plants, sort of the average isotopic composition. But I'm instead going to be talking about very specifically the isotopes from these plant wax N-alkane things. So there are a couple of bits of background to get out the way here first. So firstly, what the heck are plant wax N-alkanes? Why do we care about their carbon isotopes? And also, why is there a geologist working with BIFoR at all? It's got trees, not rocks. So to cover the last point first, I am specifically a paleoclimatologist and I'm interested in reconstructing past climates. So I'm really hoping there are curtains in here, but it might look like this outside right now. But if we were going to go back in time, maybe 100,000 years in this spot, it might look something a bit more like this, so rather more snow and mammoths than we're used to. And then if you were going to go back more like 50 million years, you might get a tropical rainforest about here. So clearly, we've got some really large-scale shifts going on in the global climate system over geologic time. And we can look at these shifts and figure out what causes them and what response from ecosystems you get. And we can use it to ask some